Hello, everyone, and welcome to this Cell and Gene Live Major Trends in Next-Gen RNA Engineering. I'm your host and moderator, as Miles said, Erin Harris, and my expert panelists for today are Sam Deutch, EVP Research and Early Development at Nutcracker Therapeutics, and Nathaniel Wang, CEO and co-founder at Replicate Bioscience. We're going to get started in just a minute, but before we do, I want to encourage you to submit your questions for our experts via the Q&A tool. We will be responding to your questions, so please submit them as you think of them. We will definitely get to them. All right, so let's get started. So nearly half of all the advanced therapy market value in 2022 came from RNA therapies. And RNA drugs have recently come into focus following the approvals and emergency use authorizations, of course, of the mRNA vaccines for COVID-19. And advancements in purification and cellular delivery of RNA have enabled the development of RNA-based therapeutics for a broad array of applications. Now, prior to the pandemic, RNA treatments were focused on rare diseases with small target markets. Now they are in clinical development to treat many more common ailments, including cancer and cardiovascular disease. And RNA therapies are cost-effective and can be readily manufactured. Now, before we get into our formal discussion with our panelists, I'd like to take a brief second to have them each tell you about their companies to kind of level set our conversation. So, Sam, I'd like to start with you. Please tell us a little bit about Nutcracker Therapeutics.
Hi, good morning, Erin, and thanks for the opportunity to participate in this very exciting panel. So Nutcracker Therapeutics is an mRNA therapeutics company that was founded in 2018. We currently have about 150 employees, and really our main goal is to develop a pretty complete and comprehensive end-to-end platform for mRNA therapeutics. You know, that starts from the manufacturing side. So we have our microfluidics-based manufacturing technology that proceeds in kind of 3 steps with 3 different biochips that get run sequentially, not at the same time. So we start with a process to manufacture the DNA template. Once that material is released after a number of QC assays, then that goes as a major input for our second process, which is the mRNA drug substance manufacturing and purification. That also happens entirely inside our biochips. Similar to the DNA template, that the material gets exported, and we run about 15 to 20 different assays to look at different aspects of the quality attributes of the material. And then one or multiple mRNAs become the input to the third biochip, which is where we perform the formulation of the RNA into nanoparticles, which are actually as you all know, the active product. Other aspects of our technology include our unique nutshell delivery technologies. This is our library of delivery vehicles. We also put a lot of effort and a lot of focus on how we design our mRNAs. We believe that this is essential for accelerating therapeutic development, and we need to optimize both the performance of the RNA, but also a less discussed aspect of mRNA, which is its manufacturability. Not all mRNAs will be equally manufacturable and lead to the same kind of quality profiles. So if you really want to accelerate your path from concept to lead selection, thinking upstream about manufacturability is very important. We are also at Nutcracker working on the development of novel mRNA formats, not only linear mRNAs. And then lastly, our platform also includes a number of mRNA and protein design elements that provide differentiation for the different therapeutic areas that we're pursuing. And these include applications in oncology, as well as in the rare disease space currently.
Good. Okay. Thanks, Sam. That's really helpful. Nathaniel, I want to give you an opportunity to tell us a little bit about Replicate Bioscience.
Yeah, absolutely. And Erin, thank you for having me on this panel. It's very exciting to be able to discuss this topic with you and Sam. A little bit about myself. I'm an immunologist and a scientist by training. Replicate Bioscience is a company that's based in San Diego. We have about 20 people now, and we're focused on a technology called self-replicating RNAs. And we believe they can really be a best-in-class type of technology, and that's based on the fact that they have a much higher bioactivity which would enable entirely new applications. So right now there are some limitations to linear mRNA, and we think we can get past that into a lot of other therapeutic areas and really bring the promise of the technology to billions more than what the technology has already delivered on. I think that something that Sam and I probably agree on is the fact that innovation without the ability to deploy those innovations tends to cause stagnation in technologies. And so we are also focused on end-to-end capabilities in terms of building our pipeline. And, along those lines, we're going to be entering the clinic imminently in infectious diseases as kind of a definitive platform demonstration of our technology, and really show that the technology can be best in class. We chose infectious disease because it was the most validated use of the technology to date, as I think the audience is probably well familiar with. And then we'll expand the technology into areas like oncology early next year, initiating a phase 1/2 trial in the space, as well as in the area of protein drug replacement, using your own bodily cells to produce a protein in situ in areas like immunology and inflammation. So a lot of exciting times coming up, and looking forward to discussing some of the engineering that led to all of that.
Good. Excuse me. Thank you, Nathaniel. And I'd actually like to keep you at the mic if you could, because we're going to jump right into the two major trends developing in the next generation RNA engineering space. So I want to start first with tissue specificity. So Nathaniel, talk us through, how can we more accurately target tissues to reduce off-target effects or to cross a therapeutic threshold for the protein expression. So what are your thoughts on that?
Absolutely. And maybe I'll start by taking a little step back on why people might be interested in that in the first place. So with conventional linear mRNA, which is the technology behind the current COVID-19 vaccines, you end up getting a small burst of protein, and then it rapidly goes away. And so that limits the types of proteins that you can really encode into RNA without eliciting side effects that are going to be unacceptable from a clinical perspective. And so you're kind of confined to proteins where you only need a small amount to be able to get that therapeutic effect. And so one of the ways that people wanted to extend on the technology is through tissue specificity. So the idea was that if we can get that small burst of protein in just the right tissue, then we can administer systemically, whether it's subcutaneously, intravenously, and it won't cause those side effects, and it will only be expressed in a certain tissue. So in all cases, it's the same type of protein profile you're talking about. Again, just small bursts are going to be okay, or frequent dosing is going to be okay, but it allows for more systemic administration and organ-specific effects. And so, the two major ways that I would say that people are going after that is first, through the way that you're actually delivering the material. That can be lipid nanoparticles, that can be polymeric particles, and people oftentimes just alter the composition of those. So the little fat bubbles, or the LNPs that encapsulate the mRNA that's used in the COVID-19 vaccines, sometimes people add a lipid or another fat component to that to help with the targeting to certain tissue types, and that's been successful. Or other times they'll just change the relative ratios or some of the individual components to have that particular effect. In other cases, people will focus on the payload itself, things like the RNA itself, and encode things like microRNAs that make them more tissue specific, or alter the sequence through different types of optimization that make it so it's more likely to express in certain tissue types like tumor tissue. So that's just a couple of examples, but I'm sure that Sam has more ideas.
Thanks, Nathaniel. That was great. Yeah, Sam, before we move on to the next major trends, what are your thoughts on tissue specificity and how we can more accurately target tissues?
Yeah, I essentially agree with everything that Nathaniel said. In our experience, that basically two main ways of doing this, through delivery and through RNA design. I think first on the delivery side, when there's a strong dependency on the route of administration. If one delivers the formulation, for for example, IV, then the first thing that happens to these nanoparticles is that they encounter the set of proteins that are in serum. And these interactions between nanoparticles and serum proteins really govern to a large extent where those nanoparticles are going to be distributed to. So one classic example is ApoE. So if your nanoparticles, after injection, they get coated by ApoE, then they will tend to go to the liver and be uptaken by cells through mechanisms that use this LDL receptor. So one way that, as Nathaniel mentioned, that people have tricks that people have used to tweak a little bit the biodistribution of the nanoparticles is by changing the charge of the nanoparticles. So when you change the charge, you actually change a little bit the interaction of the nanoparticles with the proteins in the serum. And what we have seen very clearly, and others have reported on this as well, is that when these nanoparticles have a neutral charge, they tend to go to the liver. When they become positively charged, they tend to bypass the liver and be distributed in the lung. And when they're negatively charged, the tropism becomes more towards the spleen. I think that in addition to the biodistribution, we can also think about different strategies for being more active in how we target the nanoparticles. And the approach and the thinking that we're pursuing in Nutcracker is to think of targeting through avidity rather than through high affinity interactions. And the way we're doing this is that we have our nutshell delivery technology, a Nutcracker, which is a large and highly diverse and highly tunable library of cationic lipids that we synthesize ourselves, and we have a lot of control over that synthesis. It has properties of lipids and properties of polymers as well, and so we can control the PK, the lipophilicity, and we can also conjugate a large number of different and diverse chemistries onto our scaffolds. And this allows us to explore the targeting question by adding this low affinity moieties, but that complexively they do very significantly change the avidity of the particles towards different parts of the body. And the other thing that we think about quite deeply is in controlling how these targeting groups are actually anchored on the surface of the particles. So how are they presented rather than just being randomly inside the nanoparticle in an uncontrolled way. And this is quite different from approaches that have been reported in the literature that are more around adding this high affinity antibodies that in our experience they're actually a little bit too big and end up altering the behavior of the particle as a whole, and also are quite complicated from CMC and regulatory perspective. Just quickly on the other aspect of this is on the sequence engineering side. Nathaniel already mentioned, that one approach that people have taken is engineering of the UTRs to engineer specific UTR mRNA interactions. But we are also currently in a large effort to optimize the interplay between the structure of the RNA and natural RNA binding molecules. And these RNA binding molecules, they're differentially expressed in different cell types. So you can that way engineer different behavior in different tissues or different cells. So that's using like natural interactions, but there's also engineering of novel RNA protein interactions through the use of synthetic aptamers, and we and others have shown some examples of how this is possible. For example, in the recruitment of translational initiation factors. Again, some of these are differentially expressed between cells. In the case of circular RNAs, the iris component that drives translational initiation also have cell type specific performance. So some irises will work better with different cell types. So there are opportunities there to limit off-target effects. But as we move forward, I'm very confident that we'll see more creative ways in which we can use the information that we gather from cell type specific transcriptomics to engineer desired behaviors of our mRNA therapeutics. But this is still at a very early stage.
So I had a question about something you had just brought up, Sam. Erin, I don't know whether you mind if I follow on with
No, go right ahead.
So It was interesting when you were talking about the route of administration being pretty important in terms of the biodistribution. I think that sometimes I, I've encountered when people hear tissue specificity, they hear something like kidney specificity, and they just assume that all of the material goes into the kidney as soon as you inject it. But I think those of us in the field probably feel pretty differently about what actually happens to the material. I mean, how do you all think about it?
No, I mean, I think again, when you deliver through ID, for example, I think that the first challenge for most people is to think about how are these nanoparticles going to clear the liver. A lot of things will just tend to get stuck in the liver pretty fast. So that's kind of the first challenge, and I think that's where playing with the charge of the nanoparticle is very important. And then the next thing is when, I mean, as you said, the particles won't go straight into the liver, right? So, but how can you get them so that they're uptaken in the liver with more that being a more common event than going to other parts in the body. And also the thing that we find very interesting is that localization and expression very often they don't correlate very well. So for example, when we look at tissues that we analyze independently for the presence of our nanoparticles, for the presence of the RNAs, we see quite clearly that it's not where the particles are localized necessarily that they're expressed. I think cellular uptake and then expression, release from the endosomes, all these very complex steps that we are all beginning to understand. We know play a role in terms of like the not necessarily the physical biodistribution, but the active functional biodistribution of the molecules.
Yeah, no, and I would just like to highlight that point from our perspective as well, because we agree completely that where the material ends up versus where it's expressed can be very different. And, the safety and the biodistribution of these tissue-specific approaches is a lot more complex than I think, it sounds on the surface. So just wanted to highlight that from our perspective as well.
And very surprising where we end up finding some of these molecules, which then, once we get into the safety aspects of this, I think there's more discussion to be had on those topics.
Good, thank you both, and thank you for the added value to that tissue specificity portion of this. That's really helpful for our listeners today. I want to move on to one of the other major trends, which we know to be potency and durability. So I'd like to hear from both of you about, how we can improve the amount, quality, and even the longevity of expressed protein to expand the use cases for RNA. So Nathaniel, can we start with you on that?
Yeah, absolutely. So, before I had been talking about, that quick burst of protein. And when you look at how long linear mRNAs express, they express actively, meaning they'll be actively producing the protein that's being encoded for about 2 to 4 days before that instruction manual ends up falling apart in the cell and it can no longer produce more protein. If you look at some of these new technologies, and I know that Sam had mentioned circular RNA, it represents a marginal increase on this. So you end up having expression actively for about 4 to 7 days. So, about a 1 to 3 day gain in terms of protein expression. And that's all based off of published literature out there. There's also technologies out there, and we work on one of these called self-replicating or self-amplifying RNAs. And these will actively express protein out to 49 plus days. Depending on the exact protein that you're encoding. And that's also based off of published literature out there. And so you can get a wide distribution looking at these technologies on the total amount of protein that can be produced on them and the durability for them. And that starts to open up a lot of applications. Part of the limitations of low durability and low amounts of protein is you either need to frequently redose, which can be a problem if you're having too high of a side effect profile, or you just don't get enough protein to be bioavailable to have the therapeutic effect that you need. You may need proteins expressed at higher levels, things that may soak up inflammatory molecules in the case of autoimmune disease, for instance. And you don't want to be dosing the patients daily. ideally you'd be dosing them monthly or every few months. And so developing these technologies that allow expansion of RNA tech into these other spaces are going to be things that are pretty important. I think that we also see similar trends in oncology where the current versions of the technology are being deployed in more personalized approaches, which tend to be much more expensive, as hundreds of thousands of dollars, as opposed to more off-the-shelf approaches, which would be more single digit or tens of dollars, which obviously would be more of a win for patients in terms of the actual cost of goods. So those are the broad strokes of how I see how this technology can really expand on these different areas, but I'm sure Sam has even better ideas.
I was going to ask you, thank you for that, Nathaniel. That's very helpful. And yeah, the difference in cost of goods is remarkably different, notable for sure. And Sam, I'd love to hear from you about the potency and durability aspect of, the use cases for RNA. What are your thoughts?
Yeah, I think the first thing that we think of is what is the indication, the target, and we think about of the drug first of all with the TPP. What's the target profile? How would we like to for this drug to be used in the clinic? What's the competitive landscape? What are alternative treatments that have been pursued, and then how could an mRNA product be competitive and actually be usable in the real world? There are settings that are a little bit more acute. For example, in later stage oncology, where we believe that it's unlikely that the treatment duration is going to be very long, so interventions are going to be probably like 3 or 4 doses that can be spaced at intervals of a few weeks, one from another, versus the more chronic type of indications where, for example, in the setting of a rare disease, you need to think, okay, if this works, probably the patients are going to be being exposed to the product for the rest of their lives, right? And then you think about those two things very differently, both in terms of how you design the RNA and how you design the nanoparticle, so that the safety profile of is compatible to for its use. I think in terms of potency and PK of the mRNA. At Nutcracker, we are quite aggressively pursuing two partially overlapping strategies. One is around the development of a proprietary circular RNA platform, and the second is around PK optimization by a very intentional secondary structure evolution. So both actually secondary as well as tertiary structure of the RNA, and this new concept of RNA knots or superfolding are very high interest to us, as we're seeing that these are efficient and quite powerful ways of modulating the durability of the RNA. So we're seeing that even with linear RNAs, we can see pretty significant shifts in the duration of expression, and it's not so much necessarily how long that you can detect expression from a linear RNA, but it's what that expression profile looks like, right? So you start with expression of a certain level, and then what we kind of measure is how fast that expression level drops, because ultimately what we're most interested in is the total exposure of the drug, so what that area under the curve. We see that in many real life applications, actually, you want to achieve the opposite. You want to have a very slow ramp rate of expression, and actually that time between dosing and the time that you reach Cmax is actually very, very, very important in terms of how we modulate like toxicity. For example, if you start with very high expression, you very often trigger things like cytokine release syndrome and things like that. Whereas if you drive that expression to be more gradual and slow, to the point where you reach Cmax and you can hold that expression level for a number of days, then you're in a space where your drugs can be safer but also very effective. We also think about in terms of how we deploy RNAs, whether the indication needs to be like cell autonomous versus non-cell autonomous indications. So for example, things like reprogramming or in vivo CAR Ts are situations where you need that RNA in the same cell to be expressed for some time. So applications that require the cell autonomous behavior can only be practiced in our opinion. So the mechanism of action will only be supported once your RNA durability reaches a certain threshold. In other indications, for example, enzyme replacement, you don't need the exact same cells to be producing the protein. So in that situation, even some shorter-lived RNAs that can be redosed do support the mechanism of action. But even in those settings where you're relying on systemic protein expression, you do need some amount of durability so that the product can be applied in the clinic in a competitive way. So I agree with Nathaniel that you're looking at a few weeks to a few months interval of dosing is important. Actually, also the how you engineer the protein to drive the protein PK is a very important aspect of this as well. So it's not only the mRNA, but the protein. So if you can reach a certain titer, but then you get your proteins to be active for a longer period of time. So we think of the interplay between those two things as well.
And if I might, I also wanted to add in on the subject of potency, which can sometimes be a confusing term on what that always means. And, I think the way that we've been using it is in terms of bioactivity of the molecules. But to give some sense of scale for people who might be listening in, with some of the newer technologies, they're starting to come online and will be going clinical soon. The preclinical or the animal models show that you can start to dose these things anywhere from 1,000th of the dose to 1,000,000th of the dose of what people have currently been using. And even if we look at the lower end of that, at what 1,000th of a dose would translate into and something that people might better understand, if you look at, the COVID-19 vaccines that many of us got, if you went at 1,000th the dose, then a 1 liter bioreactor or, half of a 2 liter bottle of soda could basically produce 50 million doses of a vaccine candidate, which obviously would have been transformational in terms of deployment and how we could accelerate getting technologies out to the public in those cases. So, I think that driving these technologies in the way that both Sam and I have been talking about, really opens up the avenues, not just for new products, but also in some of the existing ones as well.